Alright. Assalamu alaikum, everyone. I'm Farah Abul. I'm a fourth year medical student, and I am very, very honored to be giving my first class session of the year, and hopefully the first of many, based on you guys' requests. For today, we're going to be covering gram-positive bacteria, which is honestly one of my favorite topics. I really love microbiology. It's my favorite discipline. You guys have probably seen my immuno crash course. That was just my way of helping with micro is really where I feel at home. And gram-positive bacteria are my favorite to discuss. So hopefully through this, you guys will also enjoy it, inshallah. Is it glitching for anyone else? It says in the chat. Oh, okay, my internet connection is unstable. Just a second, I apologize. But
Uh, is this going to be just gram positive or like everything, basically gram negative too and stuff like that?
no, for this session, it's just yeah, it's gram-positive too as well, but it's just gram-positives and bacterial genetics because you guys requested that lecture as well. And I understand it's a bit tough. Those lectures aren't the easiest to navigate through. Even though the topics themselves are simple, it's just the way that the slides are constructed can be somewhat difficult. So I'll be giving you guys bacterial genetics as well. Does that answer your question? Okay, great. All right. So just in case it wasn't caught in the recording, going back, Assalamu alaikum everyone. I'm a fourth year medical student and I'm very honored to be having my first class session with you guys of the year today. For today, we're going to be covering gram-positive bacteria, which is one of my absolute favorite topics. Alongside it, we're going to be covering bacterial genetics and lastly finishing off with a few practice questions just so you guys get a general idea about what the questions you might face in the exam are like. Okay, starting off with a general introduction. Bacteria are microscopic living organisms that only have one cell. You guys obviously know all of this information. We have harmful bacteria as well as harmless bacteria that is just part of the normal flora within the body or the microbiome of the body that essentially is there to protect us from harmful bacteria by occupying the space that this bacteria might otherwise use. The way that I can identify and name bacteria depends on the shape, the color, the pathogenesis, amongst many other specific structures, let's say, that could be present within that specific bacterium. The way that I actually present the name is that I'm going to have the genus followed by the species, and they should be italicized, okay? So that I have the capital letter, as you can see here, capital letter, small letter, and those two should be italicized. I do remember having this italicized, but it's fine. All right. Taking a closer look at a bacterial cell, we can see that it is composed of a lot of different things. Starting from the outside inwards, we have this capsule, okay? Now, something to keep in mind is that a capsule is not always present. And this is very, very important because when a capsule is present, it usually has very potent effects on the type of presentation that I'm going to be expecting out of this bacterium. For example, if I have a capsule, this bacteria is going to be more likely to be capable of evading the immune system. In that sense, it's anti-phagocytic, right? So the immune system will not be able to phagocytose it. Similarly, it might induce an extreme inflammatory response, which kind of gets the body occupied with itself to the point that it just negates the bacterial cell entirely. A capsule is also very beneficial in taking a bacterium into places and sites that it wouldn't really be in otherwise. So whenever you think of a capsulated or an encapsulated bacterium, think that it is likely going to be causing something within the central nervous system, okay? Not always, but this is just a good rule to have in general, where if I'm thinking of something that can cause inflammation to those layers around the brain, surrounding the brain, called the meninges. So inflammation of the meninges, also known as meningitis, is typically caused by encapsulated organisms. Also, if I'm going towards harsher places within the body, like for example, the very, very low lobes within the lung, I'm also typically going to be discussing encapsulated organisms. The reason why I'm going into all of those details is my love for microbiology
Mhm.
does not come from memorization whatsoever. Is there a question? Okay. My love for microbiology does not come from the fact that you just have to memorize a lot of details. It comes from the fact that you can often make sense of how this particular strain of bacteria is capable of actually developing the manifestation that we see. Okay, after this capsule that is not present in all bacteria, we have our cell wall. The cell wall is present in both gram-positives, gram-negatives, but it is missing in something called mycoplasma. Okay? Mycoplasma is this acid-fast type of strain of bacteria, which essentially means that I will not be able to stain it using gram stain. Okay? The premise of a gram stain is that it wants to bind to the cell wall, right? If I don't have it at all, I'm going to have to switch to other methods in order to be able to visualize this type of bacteria. Mycoplasma is a prime example of that, and that includes Mycobacterium tuberculosis, which you guys already know causes tuberculosis or TB. Okay, the cell wall is composed of a peptidoglycan layer, which is proteins and polysaccharides that form a strong meshwork around the bacterial cytoplasmic membrane. Its functions include giving the cell its shape, resisting osmotic pressure, and it even plays essential role in cell division and cell wall biosynthesis. Given that sometimes we do not have this capsule, the cell wall is essentially going to be the outermost layer to protect from harmful and harsh chemicals in the environment, like antibiotics, right? We're looking at everything from the bacterial point of view here. We think, okay, we want to give antibiotics to kill this bacteria, but at the end of the day, to the bacteria, this is a harmful substance that the cell wall is going to try its best to fight against. After the cell wall, we go inwards to the plasma membrane or the cell membrane. This is a gel-like matrix composed of a lipid bilayer, similar to the human cell membrane. It is the most important carrier of what is called the penicillin binding protein. This penicillin binding protein is the site of action of all of those beta-lactam antibiotics that you guys have studied, all right? And I'm sure you guys know this a lot better than I do. Almost all types of bacteria have it. So when you administer a drug such as penicillin, it's going to inhibit cell wall growth and it's going to inhibit the cell membrane growth and it's eventually going to affect the bacterial cell growth in general. Okay, now we've reached the inside of this bacterium. We can see that we have cytoplasm, we have the typical structures that you're going to be seeing, as well as a nucleoid structure, which is the chromosome, and a plasmid. And those, just keep them in mind for now. We're going to get to them once we discuss bacterial genetics. Now, we have those projecting structures that come out of the cell wall, the cell membrane, the capsule. We can see them projecting outwards. Those are the bacterial appendages. We have the flagellum for movement, the pili, which are those longer extensions. They're not quite as long as the flagella, and they're not quite as short as those other structures that I'll get to in a second. These are called the pili, pilus for singular. These are essentially for the exchange of genetic information between generations of bacteria. Lastly, we have those smaller structures, which are the fimbriae. Fimbriae are mainly for adherence. Think of it somewhat like Velcro, right? You have those strokes going one way and then the other brush strokes going the other way. Once you put them together, they're going to adhere a lot more firmly. So fimbriae function in that sense that they're going to cause bacterial adherence. Now, we spoke about all those bacterial components. And I also mentioned to you guys very briefly gram staining. What really differentiates between a gram-negative versus a gram-positive bacterium? That is the outer membrane. The outer membrane is an asymmetrical bilayer structure that is only present in gram-negative bacteria external to the cell wall. So as you can see here, similar to the cytoplasmic membrane, which is this lipid bilayer, we're going to have an outer membrane in gram-negative organisms. Now, why is this important? What I want you guys to imagine is that the purple dye that I want to use really loves the letter P, okay? So purple, positive peptidoglycan. This purple dye wants to bind so desperately to the peptidoglycan wall. If I have an outer membrane in the way preventing this binding, then I simply will not be able to stain the cell with this purple coloring. Because of that, the cell is going to be stained with the previous steps that I used, which is going to be pink, okay? So a gram-negative cell, due to the presence of this outer membrane, is going to give me a pink color on microscopy. Meanwhile, a gram-positive cell, due to its very thick peptidoglycan layer, it needs it to be thick due to the absence of an outer membrane and wanting to protect itself, is going to have this thick peptidoglycan layer and it's going to bind very strongly to the purple dye and give me a purple color on microscopy. Now, is the outer membrane the only difference that we can take note of between gram-negatives and gram-positives? No. We can also see that we have those projections coming out of the outer membrane called lipopolysaccharides, or LPS. Those are essentially going to be virulence factors, and they're going to be part of the pathogenicity of this bacterium, and they're going to help and aid in the process of this bacterium to infect a cell. So for almost all, if not all, gram-negative organisms, you're going to see that one of its virulence factors is LPS, lipopolysaccharide. And if it's not mentioned, then it's just probably assumed to be there. Okay? All right. So now we spoke of a classification where we have either a pink colored bacteria or a purple colored bacteria. What other type of classification can I use in order to determine what type of organism I'm dealing with? Oxygen tolerance. Oxygen tolerance is essentially the ability of a bacterium to deal with the oxygen, depending on the presence of particular enzymes that can kind of digest and utilize that oxygen. So when I am referring to obligate aerobes, obligate meaning necessary and aerobes meaning they require oxygen. So if I were to put them in a flask with a loose fitting cap, where I'm allowing some kind of oxygen from the environment to leak into the flask, you can see that obligate aerobes float all the way to the top, trying desperately to fetch any type of oxygen that they can. Okay? Their ability to use and utilize that oxygen is because of three main enzymes: superoxide dismutase, peroxidase, and catalase. Okay? Now, going into the opposite end of the spectrum, we have obligate anaerobes. Obligate meaning necessary, and anaerobes meaning no oxygen. So these are not able to survive with any oxygen whatsoever. Why is that? Anaerobic bacteria, they cannot process oxygen because they lack a cytochrome system. From pharma, and I believe even from Mole last year, you guys do know the cytochrome P450 structure, which is essentially utilized for free radicals. It's used for drug metabolism. It's used for oxygen. It's used for a lot of different types of metabolism. In obligate anaerobes, they lack a cytochrome P450 system. They lack cytochromes in general. And because of that, they are unable to process those reactive oxygen species that end up occurring, and ultimately they would die. Obligate anaerobes cannot survive in the presence of oxygen because not just do they lack those cytochromes that I told you guys about, but they also lack the enzymes that we spoke about, the superoxide dismutase, the catalase, and the peroxidase. Now, moving on to two types that essentially can tolerate oxygen, but they're not the biggest fans of it. Facultative anaerobes, essentially, they kind of prefer to have oxygen, but they can survive without it. Aerol-tolerant anaerobes are anaerobes that can tolerate a lack of oxygen. So air-tolerant anaerobes, they don't need oxygen to grow, but they can tolerate it because they have small amounts of superoxide dismutase and peroxidase, but they lack catalase. Okay, and that's why they're not able to fully process and fully digest that oxygen. Okay, now looking at those different types of bacterium here, we can see obligate aerobes, example of that is Pseudomonas. Obligate anaerobes, an example of that is Bacteroides. Facultative anaerobes, E. coli and Strep pyogenes. Aerotolerant is like Campylobacter. Out of these, the ones we will be discussing today are Strep pyogenes and Campylobacter. So just keep those two in mind. Okay. Now that I know gram positive, gram negative, I know that they can be tolerant to oxygen in varying degrees and varying levels. How do I actually get to see all of these organisms? How am I able to properly assess what type of organism I have? After I take a culture, after I take a swab, after I take a biopsy, after I take whatever sample it is from my patient, I need to do something called culturing. Okay? Now, culturing can be done on various different media. What that means is it can be liquid, it can be just a transport media, which you basically use until you reach the lab. But the ones I really want us to focus on are solid culture media. Solid culture media are essentially liquid media that had agar added to them so that they solidify into this jelly-like consistency. Okay? They have complex constituents like chemically digested proteins, and they have inhibitory substances, and they have a bunch of different things that are helpful so that we can grow this bacteria. But we need to be able to determine which culture media I need to use for which strain of bacteria. And that comes with clinical practice. If a patient is presenting to you with a specific manifestation, you're going to test for the most common etiological agent for that manifestation and for that clinical presentation. Now, the types of solid culture media can be broadly categorized into basic, enriched, selective, and differential. From the name basic, this is just your standard media. There's nothing really special. It just gives the very basic things that a bacteria needs in order to survive. Okay, this is a nutrient agar. It's just giving it nutrients, okay? This is used when we basically just want to keep it alive for a bit. It's not really going to help much with us determining the type of bacteria, or we want to like increase the sample size that we have because the very small amount that we had originally was not sufficient for us to make a diagnosis. So we'd use a basic type of media. Okay. Now, enriched media. From the name, what do you guys think this means? Enriched, okay, it means it possibly has added nutrients. Sure, but that what makes it different than a basic one then? Enriched has particular nutrients and complex constituents like blood lysed proteins, vitamins, growth factors, extracts of yeast or meat in order to encourage the growth of particular bacteria that can otherwise not grow whatsoever. Okay? So I want you guys to think that bacteria prefers to grow inside the human body. And enriched media is essentially trying to compensate for the lack of being inside the human body by giving bodily components into that media. Okay? Next is a selective media. What, from the name again, selective means that, okay, it's going to be super specific, right? It's very picky. So a selective culture media is only going to permit the growth of a specific type of bacteria while preventing the growth of other types of bacteria. The way that it does that is that it's going to have inhibitory substances like bile salts, antibiotics, dyes, chemicals, you name it. The whole point is, if I want to grow a sample of Staph aureus, for example, I don't want my culture to be contaminated with E. coli. I don't want to just randomly see strains of Pseudomonas, right? So I want to be super selective, and I'm only going to provide what is necessary for Staph aureus to grow, given that that is the common etiological agent that I have in mind for my patient's case. Next, we have a differential culture media. Once again, from the name, differential meaning that it is going to try to give you a diagnosis, right? This is even seen grossly. I don't need a microscope for this. A differential media is one that changes and is recognized based on colony color. Okay? It's going to give you distinct morphological features based on the growth and the interaction that that bacterium has with the culture media. Okay? So let's take examples of those. The first one that we're going to discuss, and I'm not going to go to the next slide because I want you guys to tell me, is something called a blood agar. Now, what do you guys think the type of media is? Keep in mind that it's not always exclusive, okay? It can be two at once, and it often is two at once. So tell me, what do you think a blood agar is?
Enriched media. Basic.
Okay, so we got basic and we got enriched. Why basic and why enriched? It's
I
okay.
answered.
There are no wrong answers. Live your best life. It's okay.
All right. I might be wrong, but I answered enriched
Hmm.
because it has blood, which the bacteria can feed on.
Okay. Why basic?
I don't have an answer صراحة بس يعني I
Fair
think
enough.
McConkey and others are more يعني complex than just blood agar, so I said
Hmm.
basic.
Okay. Sure. Blood agar is kind of the underdog, right? You don't really assume much of it. It's kind of just the answer you usually give. You're like, ah, yeah, fine, sure, do blood agar. So I understand why you'd say basic. But blood agar is actually enriched and differential. How can it possibly be differential? Let's take a look. It is enriched. I'm not sure why it's lagging. Okay, there we go. It is enriched because of the presence of blood components, as your colleague rightfully mentioned. I am providing the bacteria with bodily components, right? I'm giving it actual blood. It's not human blood, sure, but I'm giving it blood components that it can feast on. Now, why is it differential? It is differential based on the type of hemolysis that occurs or breakdown of the red blood cells. Once you actually see this type of manifestation, you can tell, okay, this isn't just a regular blood agar, right? It's not just red all the way around. Something is happening here that is abnormal. This is hemolysis, particularly beta hemolysis. So we have partial, complete, and no hemolysis whatsoever. Partial is alpha hemolysis, which gives you kind of a greenish hue. Think of how a bruise forms. Think of all of that, like the coagulation that we took, the biliverdin, the bilirubin, all of those. So you get biliverdin before you get bilirubin, right? Biliverdin gives you this greenish color. So this is partial hemolysis. Bilirubin gives you this yellowish color, which is complete hemolysis. And then lastly, if you have no hemolysis whatsoever, this is called gamma hemolysis. Okay? So alpha, beta, and gamma. With gamma hemolysis, as you can see here, I don't see, like, sure, I see the bacterial colonies growing, but surrounding them, there's nothing happening to the culture plate. Whereas here, surrounding those bacterial colonies, I can see this huge area of yellow that is my beta hemolysis. So this is very beneficial, and you're going to see why later on when we actually discuss bacteria, for us to discuss what type of presentation I can expect for this patient even. Okay, our next one is mannitol salt agar. What do you guys think it is?
Differential and selective.
Okay, great. Differential and selective. It is selective because it only allows for the growth of Staphylococcus species. Okay, so Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus epiphyticus, you're going to see those. Okay, only. It's not going to allow the growth of strep species. It's not going to allow the growth of E. coli. It's not going to allow the growth of gram-negatives. It's very, very selective. It is differential because it includes mannitol, which is a type of sugar that can only be fermented by certain types of staph, particularly Staph aureus. When Staph aureus ferments the mannitol, it's going to turn this otherwise pink culture plate into a yellow culture plate. This is a positive mannitol salt agar, and it's going to be for Staph aureus because of the mannitol fermentation. Next up, we have the McConkey agar. McConkey agar, just like the mannitol salt agar, is also going to be selective and differential. Now, the issue here is that they both have the same colors, and I personally had a huge issue with those two because I would commonly confuse them. But the only way that kind of made sense to me was to think that, okay, it's a stretch. Okay, I'm telling you right that it's a stretch, but it helped me. McConkey is kind of like a monkey. And what do monkeys like to eat? I know it's just a stereotype, but they like to eat bananas. And usually when you have bananas, you like to have them with milk. So the monkeys are already taking up all the milk. Okay, they don't want to give you any of the milk. And bananas are yellow. So if you're not fermenting lactose because you don't have it, you're drinking all the milk, you're gonna have a yellow, like a banana type of appearance. You're not fermenting lactose. If you are fermenting lactose, the monkey isn't available, the banana isn't available, and it's going to be pink. Okay? So fermenting lactose means that it's going to be pink. Not fermenting lactose is going to be yellow. It might not make sense, but it was the way that I thought of it. Okay? That's the only way that I was able to make up the difference between McConkey and mannitol salt. Okay? So for mannitol
Can you
salt,
please repeat it?
sure. Okay. For
Thank you.
mannitol salt, we said it's going to turn yellow when mannitol is fermented. For McConkey, it's the other way around. It's going to turn pink when lactose is fermented. Now, since both of those have the exact same colors, pink and yellow, it can get very, very confusing. So the way that I remembered it is that McConkey is like a monkey. It sounds like monkey. And monkeys like bananas, which are yellow. Bananas are often also taken with milk. So if a monkey is eating his banana, he's going to take up all the milk and all the lactose, and you're not going to be left with anything. So it's going to be yellow like the banana that he's eating, and you're not going to have lactose fermentation because he's drinking up all the milk anyway. But if the monkey does not have a banana, he doesn't need to drink any milk. So you're going to have lactose available for fermentation, and it's going to be pink, if that makes sense.
Yeah, thank you.
Of course. Okay. Do you guys have any questions so far from the introduction before we get to bacterial genetics?
No questions, but would you like to take a break for a cigar?
Sure,
All right.
sure. We can come. All right. Now, for the second portion, we're going to be covering the notorious bacterial genetics. I tried my absolute best to make this as simple as humanly possible, and hopefully you guys understand it very well. Okay? In order for us to be able to actually dissect bacterial genetics and understand how bacteria is actually able to replicate, we need to understand the genetic material of bacteria and the genetic components that actually are in the process of genetic transduction, let's say, or transformation or transposition, or the various different processes that I'm going to tell you guys about soon. A bacteria has, or bacterium, sorry, has its DNA in two parts. Either the bacterial chromosome, which you guys can see here, and this is typically going to be a double-stranded DNA haploid. It's going to be the main portion that is responsible for replication within the bacterial cell when it comes to vertical type of transmission. What is that? We're going to get to that in a second. The other component of a bacterial DNA or genetic material, sorry, is the plasmid. The bacterial plasmid is a smaller part of bacterial DNA. It is extrachromosomal. It is not within this chromosome, and it exists in a single or multiple copy per cell. Okay? A plasmid is not exclusive and it is not permanent. What I mean by that is that it can very often incorporate itself into the chromosome, redistribute its genes, and then pop back out again as its own plasmid. Now, this is very important because when I am talking about two bacteria within the same generation, in order for them to be able to communicate, the way they do so is typically through plasmid rather than through the chromosome. The chromosome is pretty protected and sacred for the bacterium, so it's not going to want to just randomly exchange it. In order for me to have some kind of exchange, I'm going to utilize this plasmid, okay? Now, a plasmid can be conjugative, meaning that it is actually present within the process of this replication, or it can be non-conjugative, which we're not yet sure of the function of, okay? Now let's take a look at a deeper dissection of the functions of a plasmid. When I tell you guys that the plasmid is the one that is mainly responsible for the interactions between different bacteria, what type of functions come to mind? Well, I need to be able to reproduce, right? So I need to have some kind of fertility that is involved with plasmids. Also, if the plasmid is the one that really interacts with the surroundings, then it needs to be able to do four other functions where it can resist antibiotics, it can resist harmful chemicals in the surroundings. It should also be able to metabolize things in the surroundings in order for its survival. Thirdly, it should be able to kill nearby bacteria that it competes with for nutrients. And lastly, it should be able to cause disease, right? Because that is the whole point here. So let's take a closer look at that again. Plasmids have five main functions. Fertility, which is basically reproduction, and that is through fertility plasmids, or denoted as F plasmids. Now, F plasmids contain the TRA genes, which code for the sex pili. Remember those long fimbriae that I showed you guys previously? Those pili are involved with reproduction, and they are encoded by F plasmids, okay? Plasmids are also involved in the process of resistance through resistance or R plasmids. These R plasmids are going to contain genes that code for resistance to various antibiotics. Pretty self-explanatory. Now, why would I want to carry this resistance within a plasmid rather than the chromosome? Think about it with me here. I want to be able to kind of maintain within my entire generation a strain that is capable of resisting that bacteria or the bacterial antibiotic so that I am not fighting it alone. Okay, that's basically the premise. If the resistance plasmid, or if the resistance proteins, or if the resistance codons were within the chromosome instead, it cannot readily exchange that information unless it goes through various processes first, where it creates a plasmid in order to move that function along. Okay? Killing is basically using call plasmids, which are genes that are bactericidal and they can kill other bacteria in the surroundings. As I told you guys, if they are competing for the same nutrients, this can be very, very beneficial. Sometimes some bacteria might also have a superimposed infection, where you have the first infection and that kind of decreases the immune response or makes the host immunocompromised such that you're able to kill off that bacteria and provide a much, much stronger type of infection as well. The fourth function is metabolism, which contain genes for digestion of organic compounds and other unusual substances just to keep the bacteria alive. Last but not least is virulence. Virulence is a pathogen or a microorganism's ability to cause damage to a host. Virulence plasmids are going to contain genes for certain virulence factors and toxins that help in bacterial pathogenicity. So those virulence plasmids are very, very, very essential when it comes to me deciding, okay, is this a harmful bacterium or is it a good bacterium for the body? All right, I have my plasmids, I have my chromosome. How will I actually be able to have that gene transfer that I was telling you guys about? That is in two main ways. Either a vertical type of transmission or a horizontal type of transmission. When I am talking about vertical transmission, I am referring to the change of, sorry, not the change, the reproduction into a daughter generation. So think of your mother giving you certain things, okay? This is vertical transmission from a mother cell to a daughter cell. I'm not talking about a same generation here. Your mother and yourself are not from the same generation. So vertical transmission has this mother bacterial cell that is going to replicate itself, perfect replication, and give me daughter cells that are the exact copy of this mother cell. Whereas for horizontal transmission, I am talking about transfer of genetic material within the same generation. That's like you giving your friend your traits, you giving your batchmate your traits. Humans, it seems very foreign to us, but an easier way to imagine it is if you cough, all the people around you cough, right? You don't need to be pregnant to transmit the sickness. If you're coughing, everyone around you might also get sick. So this is horizontal transmission, okay? Binary fission is vertical transmission. Absolutely, yes, you are correct. Okay? So vertical transmission, identical daughter cell, different generations. Horizontal transmission is going to have a mother cell that, sorry, not a mother cell, it's like a sister, let's say, where it's giving into the exact same generation. Our focus is going to be all about horizontal transmission. Vertical transmission is very, very self-explanatory. Binary fission, you guys know it already. Horizontal transmission can occur in four main ways. I know this diagram only has three because those are the main three, but there's one more that I would like to discuss with you, and that is called transposition. When I am referring to transposition, what I mean is that I am able to integrate the plasmid DNA into my bacterial chromosome and vice versa. Okay, so I'm extracting a small part of the bacterial chromosome to become a plasmid, or I am reintegrating the plasmid into the bacterial chromosome. The portion of that plasmid that is actually getting exchanged is called a transposon or a jumping gene. So think P for plasmid, okay? Remember how I told you guys that if we have genes encoded on the chromosome, it's going to be very, very difficult for us to move things around. So transposition is the process whereby I am able to actually make this malleable type of bacterium that is capable of transmitting its DNA sequences into a plasmid or to reintegrate that plasmid back into the chromosomal sequences. Okay? Now, this can be very, very important because sometimes a bacterial plasmid that is received from another bacteria within the same generation might have that antibiotic resistance that I told you guys about, right? And if I were to incorporate that into the chromosome at that point, then I can ensure that through vertical transmission, I'm going to continuously have generations that are also antibiotic resistant. Okay? Is this clear? Do you guys have any questions about transposition? Okay. Moving on. Now that we
Sorry,
have,
no questions.
okay, now that we have the plasmid that we want, okay, we've given it all the desirable traits. How do I actually ensure that I am moving this plasmid into a different bacterium? That is through the process of conjugation. So remember how I told you guys that we have conjugative versus non-conjugative plasmids. This is where this comes in. If I have a conjugative plasmid, this is called an F positive bacterium, okay? Because it has the fertility plasmid that codes for the sexual pilus, okay? This sex pilus is going to receive signals, let's say, from a recipient that does not have a sex villus. It's lacking. It's called an F negative bacteria. Okay? So this F negative bacteria does not have a conjugative plasmid. It does not have an F plasmid that is able to cause it to sexually reproduce. What happens in conjugation is this donor bacterium, which is my F positive bacteria, is going to fuse with my F negative bacterium. Okay? After this linkage, it is going to create a mating bridge where it's going to make a copy of its plasmid. I'm not sure if you guys can see my pointer. I hope you can. It's going to create this mating bridge where it's going to have a copy of its plasmid transferred into this recipient bacterium that lacks the F plasmid. Okay? Once this has been transferred, we now have two cells that have this F plasmid as well as this sexual pilus, right? So we have this process where, just to say it again, we have a donor bacterium that is F positive. It has a conjugative plasmid. And we have this recipient bacterium that lacks an F plasmid. It is F negative. What happens is that this F plasmid positive bacterium is going to link with the recipient. It's going to create a mating bridge and replicate its plasmid DNA, this conjugative F positive plasmid. It's going to replicate it into the recipient such that they now both have an F plasmid. They are both F positive and they can both code for a sexual penis. Now, this new donor, which was our recipient, can go along and do the exact same process again. Is this clear?
Yes.
Okay. All right. Someone asked me to repeat transposition. Sure. Okay. So I told you that transposition, P is for plasmid. Transposition is the process of incorporating transposons, or essentially portions of a plasmid back into a chromosome, or portions of a chromosome into a plasmid. This is within the same bacterium. It's just reorganizing itself so that the plasmid is more readily available. It has everything that it needs. It's basically just perfect in its own way. Okay? So those transposons are those jumping genes that can transfer between the chromosomal DNA and the plasmid DNA. Okay? They're DNA fragments and DNA elements, but they can just shift between the two. They can either, once again, be extracted from the chromosome and incorporated into the plasmid, or the plasmid itself can get reincorporated into the chromosome, rearranged, and then produced again. Does that make sense? All right, perfect. That's already 2 down, you guys. Okay. Our next type of genetic transfer is called transformation. Now, from the F, we're going to consider free DNA. This is very, very important when I'm considering that I'm growing in a petri dish, in a culture, a bacteria that I want to create resistant strains of. Now, why might I want to do that? For research purposes, for example, sometimes even for certain drug, let's say, clinical trials. So what happens in transformation is that a dead bacterial cell, or even just the media itself that I am culturing these cells in, is going to release DNA fragments. These DNA fragments, so fragments free transformation, these DNA fragments are then going to get incorporated into a readily available bacterium. Now, can all bacteria undergo transformation? Can all bacteria actually be able to accept free DNA? No. Whether or not a bacterium is able to integrate DNA fragments depends on its competence. So transformation is sometimes also known as competence, and that is the ability of a bacterium to take naked genomes or DNA from the environment and use them. So once again, for example, if we have some media with bacteria in it and we put some raw naked DNA, which encodes for antibiotic resistance, into this medium as well, this bacteria that is competent will be able to take the DNA from the environment and use it to become antibiotic resistant. This can also be used to our advantage. If we have a very competent bacteria, what we can do is add DNA lysing agent. If I add a DNA lysing agent into the environment, this is going to degrade all those DNA fragments, and it's going to eventually get uptaken by the bacterium and degrade its own DNA as well. Transformation is very specific because only a few species are actually competent. So this is seen exclusively in Streptococcus pneumonia, Haemophilus influenza, and Neisseria species. They are very competent bacterial organisms that are able to incorporate freely floating naked genomic DNA into their own DNA to become a transformed bacteria. Is that clear?
All clear.
Perfect. Now, how were they actually able to figure all of this out? Yes.
Transformation. It should be the dead bacteria that is donating the fragments, or it can also be a live bacteria too.
Okay, so if it's an alive bacteria, it's not going to just give away DNA fragments randomly, right? But if we had in a lab setting, if we put live bacterial cells that are competent and we added DNA fragments on our own, then it's going to be the exact same process.
Basically, a dead bacteria donates fragments to an alive bacteria. That's it.
It's not that it donates it, it's just within the same environment. And if a bacteria is competent, then it's going to take up those DNA fragments through the process of transformation. Is that clear?
I do have a question.
Sure thing, go ahead.
So are these the DNA fragments will be incorporated in the chromosome or the plasmid, or it doesn't matter?
Very good question. I'm honestly not sure. I believe it could be within the chromosome, but I cannot tell. Okay. That's not something I saw, but I believe
Sorry.
it's all good. That's not something I saw anywhere, but that's a good question.
So the basic thing that fragments are in the surrounding and the bacteria will take it up. That's it.
Exactly. Yes,
Thank you.
of course. Okay. Now, how were they able to actually discover that some bacteria can transform? That was done through the Griffith experiment. And for those of you that have ever read the manga or watched the anime Berserk, you know that we hate Griffith, and this is not any different. So the Griffith experiment involved injecting a mouse with Streptococcus pneumonia, which, remember, I told you guys is a very competent bacteria that is able to actually undergo the process of transformation. So when they injected the mouse with live Streptococcal pneumonia cells, the mouse died. What they did after that was that they tried to kill those bacterial cells and see whether or not they would have an effect on the mouse. Nothing happened. Once they heat treated or they killed the strain of strep pneumonia and injected it into the mouse, nothing happened. They also noticed that a specific strain is not able to have this type of effect on the mouse, right? Where we have this strain, not necessarily for strep pneumonia, okay? We have this strain that's non-encapsulated, okay? So remember, I told you guys encapsulated means that it's going to have this greater resistance to the immune system, so on and so forth. If I were to inject a strain R, live cells, they are alive. If I were to inject the mouse with them, the mouse survives. Okay, so when I inject them with live strep pneumo cells, they die. When I inject them with dead strep pneumo cells, they survive. And when I inject them with strain R that are non-encapsulated, they also survive. Now, what would happen if I were to place this strain R non-encapsulated with my heat-treated dead Streptococcus pneumonia cells. Supposedly, neither of those is going to kill the mouse, right? We saw that heat treated does not kill the mouse and strain R does not kill the mouse. But what they noticed happens is that through the process of transformation, those heat treated dead cells are able to reactivate themselves using the living strain's DNA. It's insane. Okay. So through the process of transformation, those heat-treated dead cells, due to their high level of competency, are able to take up this living strain rDNA and then cause this death in the mouse again. And if you were to culture whatever it was that killed this mouse, you would notice that it's living cells with a capsule and it's strain S, the same one that was heat treated and killed right here. So it's not strain R that survived. Okay? Is that clear? Does that make sense?
I'm a little bit confused because if you go to like the previous slide, you said that the dead cells gives off the
Yes.
DNA to the living cell.
Yes.
And
Yes.
here I think you said the opposite. You said that the dead cell takes the DNA from the living. So
Yes.
that's confusing.
Okay. I was sure this was going to come up. The point is mainly with the competence. Okay? So over here, even in the Griffith experiment itself, if this living strain R was a competent species, then it would have just taken up the dead DNA and it would have killed the mouse itself. But this strain is not competent. What that means is that it is not able to undergo the process of transformation. Meanwhile, this strain, the S strain, is competent. It can undergo transformation. And that's what really matters here. This strain is able to take up DNA from the environment. Does that make sense? I think you're still a bit confused. So let me explain it again.
Oh wait, I said yes, it does make sense.
Okay. Okay. All right, great.
I don't know. I think you didn't like the mic didn't catch up because like I did
Yeah.
say yes, yeah.
Okay. All right, great. Okay. Now, last but not least is the process of transduction. So the way that you can kind of remember it is transduction. D is for death, because I like to associate viruses with death, even though the bacteria isn't always necessarily going to die. So transduction refers to the process where a bacteriophage, which is a bacterial virus, carrying its own DNA, will inject this DNA into a bacterium. Once a bacterium has viral DNA inserted into it, it is considered a prophage. Okay? Now, this viral DNA is integrated. We have two main outcomes that can come out of this. Either the viruses are going to continue to replicate and replicate and replicate and replicate to the point that the bacterial cell just bursts, and this is called a lytic phage, which kills the bacterial cell. Or it can be stuck in a step prior to that, where it's just replicating, but it is not causing bacterial cell lysis. And this is called a lysogenic phase. Okay? Now, it's important to note that this is not exclusive. Okay, so the actual process of transduction could involve both a lytic phage and a lysogenic phage. And we're going to see that here. The first type of transduction is called generalized transduction. Generalized transduction is typically lytic. It destroys the host cells with the release of new bacteriophages. But the main thing that I really, really, really want you guys to focus on for generalized transduction is the fact that it has packaging of pure bacterial DNA. Now, what do I mean by that? This bacteriophage comes and infects this donor bacterium. We can see that this donor bacterium has A positive and B positive chromosomes or genes. Now, after the bacteriophage incorporates its DNA and you have this huge jumbled up mess of viral and bacterial DNA all within the cell, what can happen sometimes is that when those genes are repackaged, a specific bacteriophage might not receive any viral DNA whatsoever. It can just carry a chunk of bacterial DNA. Okay? As it carries this chunk of bacterial DNA, which in this case, let's consider it to be gene B plus, it can then go to a recipient bacterium and inject it with its DNA, right? It thinks this is its DNA now. But what that does is that it's creating an entirely new strain, somewhat, okay? Because I went to an A negative bacterium. I'm providing it with this A positive pure bacterial DNA. It's not viral whatsoever. What that happens is now, okay, I have a positive gene. It's going to cross out that negative gene and give this bacterium completely new traits. Does that make sense?
Can you repeat, please?
Sure. So the process of generalized transduction is the process of the transfer and packaging of pure bacterial DNA. Okay? I think it might be helpful for you guys if I drew this out. So let me see if I can do that. Just give me a second here. Okay. Can you see my drawing on the side?
Yes.
Okay. So we have a bacteria. I'm not gonna draw all the DNA stuff because it might get confusing. That is both A positive and B positive. At this point, I might have a bacteriophage that comes and infects this bacteria, and it's going to inject its own DNA into the bacterial DNA. At this point, what happens is that the bacterial and viral DNA are both replicating. So I have A positive, B positive, A positive, B positive, and it is not getting mixed up with the viral DNA. So the viral DNA is still all the way around here. In generalized transduction, what happens is that with the formation of new bacteriophages, instead of them taking up their own DNA as they should, which is the replicated DNA, what happens is that you're going to see them. I apologize for the very hasty drawing, but you guys get the point. What you're going to see is that they are carrying the bacterial genes instead. So they do not have any of their own DNA. This is pure bacterial DNA. What happens at this stage is we have this other bacterium here that is A negative, okay? If this bacteriophage went to infect this bacterium, then it is going to give it this pure bacterial DNA that is A positive. With that, it's giving this bacterium, the orange one here, new traits, and it is now also considered A positive. Does that make sense? So generalized transduction is pure bacterial DNA. There is no viral DNA involved whatsoever. The bacteriophage just acts as a carrier from the donor bacterium to the transduced bacterium.
Yeah, it makes perfect sense. Thank you.
Of course. Okay. Now, on the other hand, we have a different kind of transduction called specialized transduction. In specialized transduction, it's the other way around, obviously, where you are going to have an incorporation of viral DNA. Exact same thing starts off where, once again, you have this viral DNA that goes into a bacterial cell. Both start to fragment and replicate. But then, once I have virion assembly, which are those offspring replicated bacteriophages, what happens is that I'm going to incorporate a mix of both the bacterial and the viral DNA. Okay? So it usually starts off lysogenic, where I'm just replicating and replicating and replicating. When the environment is suitable, it's going to switch to a lytic phase, and then specialized transduction will continue. Now, upon excision, errors are going to occur. Part of the bacterial DNA is excised by accident. It's not just viral DNA, and it's not just bacterial DNA. It's a mix of both. Okay? It's then going to release itself and go to a uninfected bacteria, inject itself there, and now it's not only injecting viral DNA, but it's also injecting bacterial DNA, right? This bacterial DNA might have very, very unique features that once again give this transduced bacteria very, very important features. Now, if the virion or the bacteriophage does not kill this cell, if the bacteriophage did not kill the bacteria, so there was no lytic phase, it will give the bacteria superpowers of a sense so that it can now produce exotoxins, okay? Because of the presence of this bacterial DNA mixed with the viral DNA, it's going to give it superpowers. It's going to be able to produce exotoxins, such as the erythrogenic toxin of strep pyogenes, the botulinum toxin of Clostridium botulinum, the cholera toxin, so on and so forth. So through specialized transduction and through the process of transduction in general, you are able to, through a carrier bacteriophage, transmit bacterial DNA from one donor bacterium to the other and give them features that they would not have had otherwise. In generalized transduction, I am transmitting purely bacterial DNA. In specialized transduction, it's a mix of both viral and bacterial DNA. That's the only difference there. And that brings us to the end of bacterial genetics. Do you guys have any questions at all? It's kind of confusing. Can you repeat? Sure. Would you like me to repeat both the types of transduction or just specialized? I am not seeing a response. Both or just specialized is the confusing part for you? Both. Okay. So as I was saying, for generalized transduction, what happens is that I have a donor bacterium that gets infected by a bacteriophage. Okay? You don't need to apologize. I'm here to help. This is the whole reason we're hosting this session. I will repeat things 1,000 times if I have to. A bacteriophage is going to inject its DNA into a donor bacterium. Okay? Let's assume that this donor bacterium has genes A plus and B plus. During the lysogenic phase, where I'm just having this consistent fragmentation and replication of DNA, I'm going to have a bunch of freely floating DNA fragments. What happens in generalized transduction is that while I am creating those virions or those new bacteriophages, I'm only going to take bacterial DNA purely, okay? Even though those are bacteriophages, they're going to ignore their own DNA entirely, and they're going to purely take bacterial DNA. So here we see a bacteriophage that took the A plus gene, and here a bacteriophage that took the B plus gene. When those bacteriophages then go on to infect another bacterium, they are going to transmit this purely bacterial DNA into this recipient bacterium, giving it new features that it did not have otherwise. The exact same process happens in transduction. The only difference is that instead of just transmitting pure bacterial DNA, it is also going to transmit viral DNA. So in this case, errors are occurring upon excision. I'm going to take bacterial DNA with viral DNA. I'm not just taking bacterial, not just viral. I'm taking both. And then I'm going to a recipient bacterial cell and transmitting both the bacterial and viral DNA into this bacterium. Now, what happens is that if this bacteria does survive, it is going to have superpowers now because it's going to have this extra gene incorporated into it that gives it an exotoxin. Okay? Exotoxins are, like I mentioned to you guys, like the diphtheria toxin, for example, the cholera toxin, the botulinum toxin, so on and so forth. Okay? Is that clear? Okay, great. Do you guys have any questions so far before we move on to the bulk of the session? I know we already took a long time, but the next part will hopefully be fun. Inshallah. I want you guys to be as interactive as possible with me, please. If you're more interactive, we're gonna get through this faster because there are certain slides where I will not move on unless you answer. So do you guys have any questions before we move along? Nope. All right. Would you guys like a break or should we just power through? It's our last section before questions. It's the gram positives.
I think we should power through.
All right. Now, moving on to the bulk of the session and why I am assuming most of you are here. Gram-positive bacteria. When I say gram-positive, immediately what pops into mind? Purple, right? So I did a gram stain on an organism and it turned out to be purple. That is where I start. I have my gram-positive bacteria. I've assumed and confirmed that it is in fact purple in color. At this point, what I want to do is look at its shape. So what types of shapes do you guys know for gram-positive bacteria or just bacteria in general?
Cocci.
Okay, perfect. Cocci or cocci, amazing. What else do we have?
Bacilli.
Bacilli, perfect, yes. And the last one is just branching filaments, which we're not going to be discussing at all. So amazing job. We have, as you rightfully mentioned, three shapes that we can see at this point. Bacilli, which are those pencil-like elongated ovoid structures. Cocci or cocci, which are those circular spherical structures. And lastly, branching filaments, which we're not going to be discussing. Firstly, I would like us to cover and mainly discuss those cocci or the cocci. Okay? I'm going to be interchangeably using both of those terms because they're both correct. When I'm talking about cocci, I'm going to want to basically assess the type of organism that I'm looking at. And the way that I do that is through a very specific test. Do you guys know what that is? Now that I have a purple spherical type of structure, I want to see, is it this or is it that? So what type of test am I going to use? Okay, that's perfectly fine. We're going to use a test called catalase. Now, catalase is a test that breaks down H2O2 or hydrogen peroxide into oxygen and water. So O2 and H2O. You can actually visibly see this reaction where you put the H2O2 and then you put the catalase on it. What's going to happen is that it starts to bubble and froth because of the oxygen being released and the water just being left behind. After I do a catalase test, if it turns out to be positive, then this is called a Staphylococcus species. Staphylococcus or Staphylococci are gram-positive bacteria arranged in grape-like clusters, as you guys can see here. They're super coiled into one another. They look like actual grapes and they are the color of purple grapes. So just imagine that. This is what I was telling you guys about. See how it gets bubbly and frothy? This is a positive catalase test that is indicative of Staphylococcus. Okay? Now, staph species in general are common causes of healthcare associated diseases or nosocomial infections. This is a term you're going to be hearing very often, so get used to it. Okay? Its virulence factors include a peptidoglycan layer, which induces an inflammatory response, teichoic acid, which regulates the cationic concentration of the cell membrane, and it basically is, remember all the things that we talked about. What does it do? It mediates adherence, it inhibits opsonization, which shameless plug, if you watch my immuno crash course, you would know all about those details. It also has adhesion factors, which are surface proteins and antigens. It has a capsule, not always, but if it does, it facilitates attachment and it inhibits chemotaxis, phagocytosis, you name it. It also has protein A, which binds to the Fc portion of IgG at the complement binding site. Once again, shameless plug, but if you guys saw my immuno crash course, you'd see that when I drew the entire complement system, you could see that it was actually the Fc rather than the Fab portion of immunoglobulins that was involved with the complement cascade, particularly in the normal type, right? Where we have the C1 QRS and all of that. So protein A is going to bind the Fc portion so that you are not able to activate complement, and this is also going to be an anti-phagocytic effect. Lastly, we have clumping factors, and this is fibronectin binding adhesion that attaches to blood clots, and it essentially traumatizes tissue that causes it to just coagulate immediately. And that brings me to the second test that you're supposed to do. Now that I know that I have a Staphylococcus species, the test that I can use in order to be able to actually determine which species within the Staphylococcus genus I have is called a coagulase test. If I do a coagulase test, which from the name means coagulation, and it is positive, i.e., the plasma that I'm looking at became a clot, then this is called Staphylococcus aureus, the most common bacteria in life. It is the answer to basically every question. It can cause anything and everything in the body. So literally any manifestation, consider Staph aureus. Anything happening in the body, consider Staph aureus. I don't care about where it is. I don't care about how severe it is. Consider Staph aureus all the time. It is insane, okay? It is absolutely everywhere. If you forget all bacteria for your life, just remember Staph aureus, okay? So Staph aureus, we said, is a staph, so it is catalase positive. It has that bubbling, and then it is also coagulase positive, which we can see here within this flask. If we have plasma and we add this Staph aureus to it, since it has coagulase, it is going to form a clot, okay? Staph aureus is the most pathogenic out of all these staph species that we will be discussing. And it produces infections ranging from localized to life-threatening. And sometimes it can actually not cause infections in and of itself, but through its toxins instead. And we'll get to that in a second. Okay? So the disease is caused either from the bacteria or because of intoxication with the toxins, or a combination of both. And that is like something that you rarely see. Usually it's an infection that predisposes. Okay? Now, what type of clinical manifestation am I expecting to see with Staph aureus? Firstly, I'm going to start to see this honey-crusted yellowish discoloration, or those kind of like pimples that are very rough and scaly on top. This is called impetigo. Then I can see this type of manifestation here, which is called septic arthritis. Now, just looking at it, you might think this is some kind of skin infection, right? There's this redness on the skin. Maybe it's like a rash of some kind. But what we're really looking at in this case is inside the joint. We have this huge inflammation that is causing this redness. It is causing this swelling of the joint space, and it is causing a lot of discomfort for the patient. It's very painful, and this is called septic arthritis. It's also going to be warm to the touch. Now, lastly, we have something called scalded skin syndrome. In scalded skin syndrome, what's going to happen is essentially the bacterial toxins are going to slough off the entire first layer of the epithelium. And it's going to have this huge effect all over the body, where you're going to notice that the skin is starting to just peel on its own, involuntarily, and it is incredibly painful and uncomfortable for your patient. So always, always, always think of Staph aureus in this case. So it's basically 4 S's: scalded skin syndrome, staph, okay? Staph aureus. Now, I keep telling you guys about toxins and toxins and toxins. What type of toxins does Staph aureus have? It has cytolytic toxins. What that means is that it can cause hemolysis, and it can attack mammalian cells, including RBCs. So for Staph aureus specifically, it is actually beta hemolytic. What that means is if you were to culture it on blood agar, what color are you expecting to see with beta hemolysis?
Clear? Clear.
Clear as in what do you mean? Like you can see
Yeah.
all the way through it?
Yeah, yes.
Exactly, yes, because it's going to have complete hemolysis, yes. But the area around this clear zone is going to be yellow. You are correct. It's just that we typically mention yellow because that's going to be the area around it. Okay?
Yes, okay.
It's also going to have an enterotoxin, which is involved with food poisoning. And what happens is that you ingest this enterotoxin, particularly in milk, potato salads, processed meats, mayonnaise, creams. Okay, so always think if I'm mentioning some kind of pastry, some kind of cream, always, always, always think Staph aureus. What happens is that the enterotoxins are directly going to affect the intestinal epithelium. They're going to stimulate the vagus nerve, and then that is going to cause a vagal reflex where I have this vomiting center in the brain just hyperstimulated. And it's like, get this out of me. Something is wrong here. And this is the enterotoxin that causes food poisoning. And usually you just vomit it out. Okay. Now, the very, very important toxin that I want to talk to you guys about is the super antigen toxin. Okay. A super antigen toxin works by that way that I told you guys about, where it's basically overwhelming the immune system. It attaches to T cell receptors, presents on MHC class 2 for antigen presenting cells, and it leads to polyclonal T cell activation with huge amounts of cytokine. So the body is actively destroying itself, right? It's trying to hit a fly with a cannon. With super antigen toxins, eventually you're going to have something called toxic shock syndrome. Okay? Toxic shock syndrome is going to be this immense release, okay, of this toxic shock syndrome toxin, the super antigen toxin, that is going to have hyperstimulation of the immune system. And it causes this very intense and immense response way beyond what actually is deserved for this bacteria. Staph aureus actually also has other virulence factors like coagulase, which we already spoke about, catalase, which we already spoke about, and it has others including lipase, beta-lactamase, which confers antibiotic resistance, hyaluronidase, DNase, nuclease, basically select all, okay? So for coagulase, it leads to prothrombin activation and coagulation. Catalase is going to break down the H2O2 into oxygen and water, and it's going to have that frothy type of appearance. Lipase and phospholipase are going to break down lipids. Hyaluronidase are going to break down connective tissue. So overall, Staph aureus is just destructive, right? It has so much happening. It has everything in its favor. But would you believe me if I told you that there are certain places within the body where Staph aureus is normal? And that is particularly within the nasopharynx, okay? Now, this isn't something that you're going to see very, very often, but it colonizes the nasopharynx, okay? And it will not cause a disease. So it's not necessarily normal, but it's not going to cause any harm within the nasopharynx. If it were, because it's an opportunistic bacteria, if it were to get the chance to go to anywhere else in the body, then it's going to absolutely cause some kind of disease. And this beta-lactamase that it has right here is very, very, very important because it facilitates a huge spectrum of antibiotic resistance. Have you guys heard of methicillin-resistant Staph aureus, MRSA? MRSA is resistant to all types of antibiotics except vancomycin, and it's very, very difficult to treat. Okay? All right. Do you guys have any questions about Staph aureus?
Nope.
Perfect. Okay, so we've mentioned that we go first catalase. Catalase positive means staph. What did we do after catalase? We did coagulase, right? So if coagulase is positive, we said that is Staph aureus. If coagulase is negative, that can be one of two species, either Staph epidermidis or Staph saprophyticus, okay? Staph epidermidis, from its name, epidermis, is normal microbiota of the skin. But what I want you guys to imagine is, let's say that during a surgery, before they were to insert a catheter, before they were to insert an IV line, before they were to insert anything, they did not clean the skin properly. What's going to happen is that this external foreign body is going to be a conduit to just carry Staph epidermidis from the skin surface into the body. And Staph epidermidis is very unique in the sense that it creates the slime onto that foreign body that facilitates adherence and antibiotic resistance because it's very, very difficult to penetrate that slime, and that is called a biofilm. So whenever you have a foreign body mentioned, that could be even like a heart valve, that could be a transplanted organ, that could be literally anything. But for transplanted organs, think more viral illnesses, so keep them aside. If I'm talking catheters, if I'm talking IV lines, if I'm talking central lines, if I'm talking any type of tube, a gastric tube, a tracheal tube, whatever it is, think of staph epidermidis. Okay, immediately think staph epidermidis. The other one that is also coagulase negative is staph saprophyticus. The only thing that you really need to know about staph saprophyticus is it very, very, very commonly causes cystitis, which is a urinary tract infection in young women. So it's very, very common. Now, how would I be able to tell the difference between staph epidermidis and staph saprophyticus, since both of them are coagulase negative? The way that I can achieve that is through an antibiotic sensitivity testing, also called a novobiocin sensitivity test. So novobiocin is a type of antibiotic that I'm going to inoculate and just place onto a dish with this cultured organism. Okay, now at this point, I know this is catalase positive, coagulase negative. I've seen that it's gram positive, cocci under the microscope. What can it possibly be? If you have this zone of inhibition around the antibiotic, meaning that the antibiotic killed the bacteria surrounding it, this is a sensitive novobiocin test, and this is indicative of staph epidermidis. If you lack the zone of inhibition, meaning that the antibiotic was not able to kill any of the surrounding bacteria, this is a resistant strain, and this is staph saprophyticus. Is that clear? Are you guys able to tell the difference between the two?
Yes.
All right, great. Now, sometimes what happens is, I know that it's catalase positive. I don't have coagulase. How am I gonna know if this is Staph aureus or Staph epidermidis, for example? I can actually check on blood agar. What did I tell you guys Staph aureus does to blood?
Beta
Beta.
hemolysis.
Absolutely, beta hemolysis. So if I were to take note of beta hemolysis, then I would start to think of Staph aureus. Not only that, but Staph aureus comes from oros, which is gold. So Staph aureus is going to create gold colonies on blood agar that you can take note of here. Okay? Those gold colonies are mainly due to what? The beta hemolysis, right? But that is what gives it its name. Staph epidermidis, on the other hand, is going to look white on blood agar. It's not going to have any hemolysis, and it's going to create those white colonies. Another way that I can differentiate between Staph aureus and Staph epidermidis, remember which agar we used that was selective for staph?
Mannitol salt?
Absolutely, yes, mashallah. Mannitol salt agar is selective for staph and differential for staph aureus, because staph epidermidis does not ferment mannitol. If it does not ferment mannitol, which color is it going to give me? Yellow or pink?
Pink.
Absolutely, yes, great job. So if it does not ferment mannitol, it does not change color, and this is staph epidermidis. If it does ferment mannitol, the media is going to turn yellow, and this is going to be staph aureus. Is everything clear? Do you guys understand all of this?
Yep.
All right, perfect. Now, I want us to review everything that I just said when it comes to tests. This is the point where I'm not going to move on from the slide unless you guys answer. We saw that we have gram-positive cocci that are catalase positive. Now we know that they are staphylococci. What do I do at this stage? What type of test am I going to do?
Coagulase.
Coagulase, absolutely. And if it is coagulase positive, what's that going to mean?
Staph aureus.
Absolutely, yes, Staph aureus. Now, if it is coagulase negative, what's that going to mean?
Epidermis.
Either. Yeah.
Or
Oh,
Or
Staphylococcus.
Yeah, saprophyticus.
Absolutely. So we have two options here, right? And how will I be able to tell the difference between the two?
Uh, Nevo-brim.
Novobiocin
Yes,
Yeah.
sensitivity.
exactly. Absolutely. Novobiocin sensitivity. If it is not sensitive, if it is resistant to novobiocin, then which organism is it going to be?
Saprophyticus.
Absolutely. And if it is sensitive, then it is going to be epidermidis. Very proud of you guys. Very, very proud. And we are done with stuff. That's it. We're done. It is like the bulk of the session, and we're done. Is staff clear?
Yes.
Clear.
All right. Now, let's look at the other end of this diagram. We said that if it is catalase positive, then that is staph. What if it's catalase negative then? This is going to be my Streptococcus species. Okay, so Streptococci are gram-positive cocci in pairs, so diplococci, or in chains, which is what you guys can see here. They are catalase negative. Notice how we don't see any of that frothy appearance that we saw with staph species. We don't see that bubbling, right? So streptococcal species, in order for me to be able to tell the difference between them, what I need to do is culture them on blood agar, okay? If I culture them on blood agar, what are the three outcomes that I might get? Alpha, beta, and gamma, right? So alpha hemolysis is what color?
Green.
Yes, beta hemolysis is
Clear. See-through.
see-through or yellow, yes. And then gamma hemolytic streptococci will have no hemolysis. So it's gonna look somewhat gray, okay? Let's take a look at that here. So here we can see this is a blood agar. Alpha hemolysis is going to give me this biliverdin type of appearance, this greenish type of appearance. Beta hemolytic streptococci, which have complete hemolysis, are going to be clear, they're going to be see-through, and they're going to have this yellow halo around them. Lastly, we have those gamma hemolytic streptococci, which have no hemolysis on blood agar. Now, for alpha hemolytic streptococci, I will start to think of strep pneumonia and strep viridans. For beta hemolytic, I will think of strep pyogenes and strep agalactiae. For gamma hemolytic, I'm going to think of strep bovis and no longer consider strep, which are the enterococci. Okay, and we'll get to them later. For alpha hemolytic streptococci, let's take it one at a time in order. For alpha hemolytic streptococci, I told you guys that it can either be strep pneumo or strep viridans. How will I be able to tell the difference between the two since they're both alpha hemolytic? The way that I do that is through an optogen test. Very similar to the novobiocin test, where I place this antibiotic disc, and if I see this zone of inhibition, the susceptibility to optogen, this is going to be my strep pneumonia. Okay, so if it is sensitive to optogen, it is strep pneumonia. If it is resistant to optogen, if it does not have a zone of inhibition, then this is going to be my strep viridans. Notice how they both have very partial hemolysis. That's because they're both alpha hemolytic. Now let's take a closer look at both of these. For strep pneumonia, it is a gram-positive cocci that looks kind of lancet-shaped and is typically arranged in diplococci. Okay, it is non-motile and it is capsulated. So when I told you guys capsulated, what am I thinking of? Hard to reach areas, right? So think of the brain, think of the lower lobes of the lung, think of the ear, and that is exactly what it causes. Pneumonia, bacteremia, which is an infection in the blood, meningitis, inflammation of the meninges, and otitis media, which is an infection of the middle ear. Can you imagine how far it has to go to get to the middle ear? So strep pneumo is going to be able to reach all of those areas because of its capsule. What other virulence factors does it have? It has peptidoglycan and teichoic acid. So what does that mean? gram-positive, right? We know that it had a very thick peptidoglycan layer. But for strep pneumo, this peptidoglycan and teichoic acid activate the alternative C pathway. What is the alternative C pathway? Evading the whole C1QRS, just going to C3 convertase, right? So this peptidoglycan and teichoic acid is going to activate the alternative C pathway or complement pathway. And why is this beneficial for me? Once again, overwhelm the immune system. Let it destroy itself. Next, we have hydrogen peroxide, which allows reactive oxygen species to cause damage. We also have pneumolysin, which binds cholesterol in the host cell membrane and lyses it. It facilitates spreading into the tissues after the destruction of the cell membrane. We also have a polysaccharide capsule, which is very, very, very important. It is the most important factor allowing the bacteria to enter the host cells. It's also antiphagocytic. We have an IgA protease, and that's important because with those IgA protease positive bacteria, what they do is that they're going to cleave IgA and they're going to break it down. And where is IgA most present? Mucosal membranes, right? So strep pneumo, if it wants to infect the lungs, it has to be able to bypass all of that huge meshwork of IgA within the mucosal membranes to get all the way down to the lungs. Okay? Luckily, though, we actually have a vaccine against strep pneumo, which is the pneumococcal conjugate vaccine, and that contains serotypes 7, 13, and 24. Okay? Is that clear? Do you guys have any questions? Strep viridans, you don't need to know any details about it, just the fact that it is alpha hemolysis and the fact that it is resistant to oxygen. That's all you really need to know. Okay, we're done with alpha hemolytic streptococci. Let's move on to beta hemolytic. I told you guys, beta hemolytic streptococci is either going to be strep agalactiae or strep pyogenes. How will I be able to tell the difference between those two? Sorry, just a second. Sorry for that. Okay, so how will I be able to tell that this is positive, sorry, if this is strep agalactiae or if it's strep pyogenes? The next slide has a typo on it, which I will correct before I send you guys the actual presentation. But the way that you do that is through a bacitracin test, not an optogen. Optogen was for alpha hemolytic, okay? Sorry about that. A bacitracin test is going to do the exact same thing, where if it is positive or if it's sensitive, it's going to mean this is strep pyogenes, okay? If I have strep pyogenes, I can also classify it as group A strep or GAS. If it is bacitracin resistant, then this is going to be strep agalactiae or group B strep. Now, why am I saying group A and group B? That's because beta hemolytic streptococci have over 20 serological groups, all the way from A to B. But A and B are the most common type in human infection, and they have further serotypes based on the proteins that they actually encode, okay? But what I want you guys to know is bacitracin sensitive, strep pyogenes, group A strep. Bacitracin resistant, strep agalactiae, group B strep. So first, let's go to group A strep. Here I wrote it correctly, thank God, bacitracin sensitive, okay? So group A streptococci, strep pyogenes, are gram-positive cocci arranged in chains. They are catalase negative because they are streptococci, beta hemolysis, and bacitracin sensitive. They are non-motile. Some of the strains are encapsulated. So once again, what am I starting to think? Maybe meningitis, something like that. Okay. And they are one of the most important human pathogens involved in suppurative diseases, systematic diseases, and immune-mediated sequelae. What do I mean by all of this? Let's take a look first at suppurative diseases. Suppurative means that essentially something is oozing, right? And producing pus. And that is exactly what I'm going to see in the clinical manifestations. Remember that honey color yellow crusted flakes that I told you guys about that have a red base? This is once again impetigo. How would I be able to tell if this is staph or strep then? They're both beta hemolytic, they both cause impetigo. What test would be the determining factor in this case?
It's not pyogenous.
It's not pyogenes if it's catalase positive, right? So if I have impetigo and I culture, both are going to be beta hemolytic on blood agar. Both are going to be gram positive. But
No,
strep is catalase negative and staph is catalase positive. They
I
both cause
meant
impetigo. Yeah.
I meant like the impetigo is not pyogenous, like it's not filled with pus.
Mm. I get what you mean, but
Uh,
my point was, how can you differentiate between impetigo caused by staph or by strep? That was my question.
okay, sorry. Yeah.
It's all good. Okay. A second clinical manifestation that we take note of is red and thickened skin over a larger area than impetigo, which is called erysipelas. Okay? Erysipelas is going to have this type of manifestation here. Immediately think this could be strep pyogenes. Lastly, we have cellulitis. Remember how I told you guys this looks a lot like the septic arthritis picture, right? But for septic arthritis, it's inside the joint. The issue is within the joint. For cellulitis, this is a skin type of manifestation. Okay? How is strep pyogenes, which is one of my absolute favorite bacteria, by the way, how is it able to achieve all of these characteristics? It has multiple virulence factors, including a cell wall antigen, lipoteichoic acid, including M protein, which is very, 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 very important. And I'll tell you why. It also has a lot of exotoxins, which we said comes from which type of gene transfer? Exotoxins? Transduction, right? With bacteriophages. So strep pyogenes is going to have pyrogenic exotoxins, which are essentially streptococcal toxic shock disease causing. You're going to have streptolysin O and streptolysin S, which are involved with cell lysis, streptokinase for lysis of clots, streptodornase for DNA, C5a peptidase. What's that going to do? Inactivate C5a component of the complement system, and hyaluronidase, which degrades connective tissue. With all of these exotoxins and these proteins and these cell wall antigens, what this bacteria is able to do is honestly very impressive. So let's follow the course of group A strep pyogenes. Usually what happens is that you have pharyngitis or impetigo or one of the very common suppurative diseases that you're going to see with strep pyogenes. After this infection, let's take pharyngitis. After this infection, in around 3 weeks, what happens is that the body starts to attack itself. Why could that possibly be? Due to that M protein that I told you guys about. There is a process of molecular mimicry where the M protein looks very similar to the actual proteins that are on the heart and on the kidney. So what happens after those antibodies are already produced to fight off group A beta hemolytic strep pyogenes infection is that I'm done with the infection. I still have those antibodies. I look at something that looks very, very similar to the M protein that I was fighting. So I'm going to attack it. This can lead to rheumatic fever, rheumatic heart disease, which is essentially just this attack towards the heart, or it can lead to inflammation within the kidney called post-streptococcal glomerulonephritis, now changed to post-infectious glomerulonephritis. Okay? So after the suppurative disease, after I have had this infection, 3 weeks later, I've already resolved the infection. It is not the bacteria itself that is causing this. This is something that's very important. It is actually the immune system that is fighting against the body itself. It's an autoimmune type of reaction that is going to cause cardiac damage, kidney damage, and just acute rheumatic fever. Okay? Something that happens due to the bacteria itself and its toxins is that it can have a huge effect on the whole body, causing a systemic disease, also known as scarlet fever, which is not pyogenic, okay? It's not the bacteria, it's the toxin of the bacteria. And it's going to have this very, very classic manifestation that you guys are going to take later on, including a strawberry tongue, so on and so forth, okay? So strep pyogenes, it can either cause suppurative disease, as simple as just having strep throat, literally just pharyngitis. 3 weeks later, if the body is immunocompromised, it can go and attack its own self, causing rheumatic heart disease, post-streptococcal glomerulonephritis, or the toxin of it could cause scarlet fever, as well as systematic diseases. Is this clear for everyone? Do you guys understand strep pyogenes well?
Yep.
Perfect. Okay. Now, moving on, we said we have our group A strep, which was sensitive to bacitracin. Now we have our group B strep, which is resistant to bacitracin, and that is our strep agalactiae. Okay? Strep agalactiae is a gram-positive cocci arranged in chains. It's a facultative anaerobe that is catalase negative, but it is positive for the CAMP or the CAMP test. Now, what is the CAMP test? Essentially, what I'm trying to do here is I'm combining two beta hemolytic organisms on a blood agar. I put a streak of Staph aureus, and then I put horizontal streaks perpendicular to it of the organism that I have. A for arrow. If it creates this arrow type of appearance, this is very indicative of strep agalactiae, and this is a positive CAMP test. Over here, we can see that if it were a different kind of organism, this is also beta hemolytic, right? So this could be strep pyogeny. It does not have this arrow type of configuration, and thus we can assume that it is not strep agalactiae, okay? So we do not only depend on the bacitracin test. The virulence factors of group B streptococci is hydrolytic enzymes that actually break down the connective tissue and a capsule that interferes with phagocytosis. Once again, I mentioned capsule. What should you immediately think of whenever I mention a capsule? Meningitis. This is particularly the case because group B strep or strep agalactiae is normal microbiota of the intestine and the vagina in many, many women. So think of pregnancy in this case. If a woman is positive for group B strep, she does not get tested, does not get treated. If she has a normal vaginal delivery, as the child is passing through the birth canal, it is going to get exposed to strep agalactiae. A child is very immunocompromised. The immune system is very primitive. There's basically nothing there. So what happens is that strep agalactiae is going to cause neonatal diseases, including meningitis, even possibly complicated into sepsis, as well as other obstetric complications. Is that clear?
Yes.
Perfect. Now, last but not least is the gamma hemolytic streptococci. Gamma hemolytic streptococci can either be strep bovis, which we're not going to be discussing, or they can be enterococci. You do not need to know the details of them. You can just know that they are cocci, okay? They are gram-positive. They are gamma hemolytic. They are normal microbiota of the gut in humans and animals. And 2 species can cause infections. Those were not mentioned in your slides, so you don't need to worry about that. But the point is that these enterococci might develop resistance to, remember that one antibiotic I told you guys we can give to MRSA? Remember what it was? Vancomycin. So enterococci can actually develop resistance to vancomycin, thus being called vancomycin resistant enterococci or VRE. Okay? And there we go.
Nice.
What?
We have to give them an antibiotics called lionized, I guess.
Linezolid, yes. Yes, you give them
Good.
linezolid. Yes,
Yes.
good. Yes, good job.
Pronouncing the drugs is kind of hard.
It's okay. Yeah, that's perfectly valid. In POD, I had no idea what anything was, so don't worry about it. But yes, good job. Like at least you know what the thing is. Absolutely. Yes, good job. Okay, let's review. So we said if we have gram-positive cocci that are catalase negative, they're going to be streptococci. What do I do at this stage? Guys, what am I gonna do at this stage now that I know I have streptococcus species, right? I have catalase negative.
Hemolysis and blood agar.
Exactly. I'm going to culture on blood agar. And through that, I'm going to get three possibilities, right? What's the first one? Alpha hemolysis, which is partial hemolysis. If I have alpha hemolysis, what am I going to do after that? Because there's two organisms, right? How am I going to differentiate between them?
It's either viridins or pyrogenes.
Okay.
No, wait.
Pyogenes? Okay.
Sorry. Pneumonia. Sorry.
Yes, it's okay. I made that mistake all the time because they both start with the letter P, so
Exactly.
I get it, I get it. Yes. So Pyogenes is the super strong one, right? So it's gonna do beta hemolysis all the way. Okay, so we have strep viridans and strep pneumo for alpha. How would I be able to tell the difference between the two?
Opticians.
Optogen test, absolutely. So if it is optician sensitive, if it's sorry, let's start with not, so it goes in order. If it's not optician sensitive, if it is optician resistant, then which bacteria is this?
Velodorian, I guess. I don't know how to pronounce it.
It's okay, you got it right. Yes, strep viridans.
Very dense, yes.
Yes, if it is optician sensitive, then it is going to be strep.
Strep pneumonia.
Absolutely. Okay. After alpha hemolysis, we discussed beta hemolysis, which is complete hemolysis. Once I have two different bacterial structures that are both beta hemolytic, what type of test do I want to do?
Camp?
No.
Okay, sure, yes, we can do camp, but camp is specific for just one of them. If I wanted to differentiate between the two,
Bacitracin.
bacitracin,
So,
absolutely. So bacitracin, if it is sensitive to bacitracin, what is that going to be? One of my favorite bacteria?
Pyogenes?
Pyrogenes. Yeah,
Pyogenes.
Pyogenes, exactly.
Yeah.
If it is not sensitive to bacitracin, that is going to be
Agalactia.
strep agalactiae. Absolutely. Strep agalactiae is the one that is going to give me a positive CAMP test. And what does that look like? It's going to give me an arrow when it gets into contact with Staph aureus. Okay?
Yes.
Perfect. Lastly, what type of hemolysis do we have? Gamma
Gamma.
hemolysis, which is no hemolysis.
Intercocci.
Exactly.
And
Yes,
bovis.
exactly. It can either be strep bovis or enterococci. You don't need to worry about them. And there you go. You guys are done with all of gram-positive cocci. You should be proud of yourselves. Lastly, we are going to be very briefly going over gram-positive bacilli. Okay? So when I am discussing gram-positive bacilli, there are two classifications that I can use. Either I can split them into aerobic and anaerobic, or spore-forming and non-spore-forming. For aerobes, we have Listeria, Bacillus, and Corynebacterium. For anaerobic, we have Clostridium. Now, with this arrangement here, all you have to do in order for it to be spore-forming versus non-spore-forming is just move Bacillus to this side. This is going to be spore-forming, so Clostridium and Bacillus. And then non-spore-forming is going to be Corynebacterium and Listeria, okay? So spore-forming means it's going to be a bit longer, and it's going to stain very, very deeply. Non-spore-forming is going to have a V or L-shaped configuration. For our case, we are only going to be discussing Clostridium and Corynebacterium genuses. Okay, for Clostridium species, we can see that they are bacilli. They are long, pencil-like ovoid structures. Most species are non-motile, okay? They are divided into four types based on pathogenicity and clinical significance. Clostridium perfringens, Clostridium tetani, Clostridium botulinum, and Clostridioides difficile, which used to be called Clostridium difficile, okay? The range of manifestations that you can see with Clostridium is very broad, and it heavily depends on the species that you are looking at. So, for Clostridium perfringens, what I'm expecting to see is gas formation, okay? Perfringens, it's going to have gas gangrenes, it's going to cause food poisoning, once again, due to, let's say, improper, like, you know, those cans that just puff up, that's going to be Clostridium perfringens right there. Again, it's going to cause cellulitis, skin infections. It's going to cause a lot of tissue infections. Okay, that's the point. That it's going to cause tissue infections. Particularly, I want you guys to remember this awful, awful picture, gas gangrenes. Secondly, we have Clostridium tetani. Clostridium tetani, from the name, is going to cause tetanus, which is essentially this spasticity that happens in the body, and they can't really do anything about it. For Clostridium botulinum, it's also going to have a spasticity, but it's going to be a different kind of paralysis that I'm going to show you guys in a second. Botulism is this intoxication due to the toxin that Clostridium botulinum has, and this can manifest in lots and lots of different ways, including even just vomiting, for example, because Clostridium botulinum can also be present in those puffed up canned goods as well as honey. Most of us don't have any issue with it at all, but you are not supposed to give it to children under 1 year of age because they will not be able to tolerate it. The tetanus toxin and the botulinum toxin are proteins involved in neurotransmission. That is why I'm going to start to expect that type of spasm, that paralysis, because it's related to the nervous system. Fourth is Clostridium difficile. Clostridium difficile is always, always, always associated with GI diseases. Particularly, something that it is known for is that if I were to give a person prolonged antibiotics, particularly clarithromycin, if I were to give them very prolonged antibiotics, what happens is that I am essentially sloughing off the normal microbiota of the GIT, which is very, very, very important. In that case, Clostridium difficile is going to make an opportunistic infection and create something called pseudomembranous colitis. Okay? So it's going to create those pseudomembranes, those filaments, let's say, that's going to be associated with inflammation of the large bowel. Okay? Last but not least is Clostridium septicum. I did not mention it in the previous slide because it's not something you commonly see. But if it is present, this is a huge red flag because this is very common with colonic malignancy and with immunosuppression. So investigate your patient for a cancer, la samahallah. Let's take a look at each of these a bit closer. Firstly, we have Clostridium tetani. Clostridium tetani is a neurotoxin that causes tetanospasm. It causes this type of manifestation here, where everything is flexed, they're having uncontrolled spasms, they don't know what to do, it's like they're having a seizure, okay? Everything is very, very, very stiff. It's called spastic paralysis, okay? This is associated with Clostridium tetani. Clostridium botulinum is on the opposite end of the spectrum, where everything becomes very floppy, very relaxed. And the reason for that is that it blocks the release of acetylcholine that is used for muscle contraction. If I don't have acetylcholine, what am I going to be able to do? Nothing, right? So I'm going to have this flaccid type of paralysis. And it's going to be a descending flaccid paralysis that starts from the head and goes all the way down to the toes, okay? Next, we have Clostridium perfringens, which I told you guys think immediately of gas gangrenes. Lastly is Clostridioides difficile, which has toxin A, which is an enterotoxin that causes the pseudomembranous colitis, and toxin B, which is a cytotoxin that disrupts the cytoskeleton, affects the cells, disrupts them to facilitate the infection with toxin A. So both of those together are going to provide me with the pseudomembranous colitis picture that I see. And that's it for the Clostridium species. You guys have any questions?
No, ma'am.
Okay. Last genus we will be talking about is Corynebacterium. Corynebacterium are small pleomorphic club shape. The way that I look at them, it kind of looks like a pot, you know, with two handles on either side that you're just carrying. That's the way that I have always seen it. So Corynebacterium is a gram-positive bacilli that appears in short chains that kind of look V-shaped. You can kind of see that here where it like has a kink in the middle, okay? Or they're gonna look Y-shaped. So don't bother too much, just know that it exists, okay? The cells have metachromatic granules on either side, which you guys can see here, that can be stained using methylene blue. It A lysogenic bacteriophage encodes for potent exotoxins in virulent strains of Corynebacterium, which we're going to be talking about very, very soon, including Corynebacterium diphtheria, which is the one we're going to be focusing on the most. Corynebacterium JPM, I've never heard of, to be completely honest, before this moment, but it causes opportunistic infections in immunocompromised patients, like patients with blood disorders, bone marrow transplants, intravenous catheters. It has a multi-antibiotic drug resistance, and it carries on the skin of hospitalized patients in about 40% of cases. So it's very, very common for it to be spread around. But as I said, it's an opportunistic infection in immunocompromised patients. So if I have an immunocompromised patient, I would not really start thinking of this type of bacteria. I'm going to mainly start thinking of a fungal agent. So don't stress yourself out too much about it. The one that you do need to know the details of, though, is Corynebacterium diphtheria. Corynebacterium diphtheria is a gram-positive rod. It's a fastidious organism. What does fastidious mean? We said that it can essentially, it prefers oxygen, but it can live without it, right? It's cultured on serum tellurite agar, which is not a culture that we've seen for anything else. So just remember it in case, okay? It has a diphtheria toxin, which is caused by a bacteriophage, and it has a vaccine, luckily, called the DTaP or Tdap. Different ones for different ages. You don't need to know the details. Corynebacterium diphtheria, when it comes to clinical manifestations, is going to be very unique when it comes to the pharynx because it's going to create the pseudo membrane that you see here, this meshwork that I'm going to be able to visualize using a tongue depressor. All other types of pharyngitis are not going to have the pseudo membrane type of configuration, okay? Something else that is going to help me determine, is this Corynebacterium diphtheria, this C. diphtheria, is it toxigenic or is it a non-toxigenic strain? Because sometimes Corynebacterium diphtheria might be harmless. It might not include the toxin, that exotoxin, the diphtheria toxin. So what I do is called an ELIC test. An ELIC test is, essentially speaking, I'm going to get a filter paper that is soaked in the diphtheria antitoxin, okay? And then I need my controls. A positive control that I know has the toxin, and a negative control that I know does not have a toxin. Both of those are Corynebacterium diphtheria, okay? One is just non-toxigenic, one is toxigenic. And then I'm going to have my actual test strain that I want to be able to determine. Is this toxigenic? Is it non-toxigenic? If it isn't toxigenic, nothing happens. It's going to look like this negative control. But if it is toxigenic, what happens is that I'm going to have this projection, this V type of appearance here, due to the antitoxin that is present. So this antitoxin is going to seep through this test strain and give me this type of formation here. Is that clear for everyone? I take that as a yes. Okay. Now, how does the diphtheria toxin actually work? It enters into the cell by attaching to a receptor and it gets endocytosed. As it gets endocytosed, it gets translocated to a region within the endosomal membrane. Okay? Once that happens, it's going to essentially inactivate an elongation factor 2 or EF2. From the name, elongation factor, can you repeat? Sure. The ELIC test. Would you like me to repeat the ELIC test? Yes. Okay, sure. Okay. So the ELIC test is used to determine if, okay, now I know that this is C. diphtheria. I know already. But I'm not sure if this strain that this patient is infected with has the exotoxin, the diphtheria toxin, or not. Because not every single Corynebacterium diphtheria is positive for the toxin. So what I want to do now is I know that it's C. diphtheria. I know. But I want to know, is it toxic or is it not? So what I do is I'm going to have a control strain here that I know is positive for the toxin. I know that this is positive. It has to have a reaction. And then I'm going to have another control that's going to be negative for the toxin. It's not going to have the toxin. Okay, so this is a Corynebacterium diphtheria strain that does not have the diphtheria toxin. Lastly, I'm going to place the sample that I took from the patient, okay, the test strain, in the middle between those two. I'm using those as my controls because if my negative control that I know is a negative control has a reaction, then I know that that test is defective or contaminated. Same for the other way around. If my positive control that I know is a positive control does not have a reaction, then that test is defective. So my control is there just to basically show me what each reaction is going to look like. I put my test strain in the middle, and then I'm going to put a filter paper that is soaked in an antitoxin for the diphtheria toxin. Okay, so it's like an antibody to the diphtheria toxin. As I put this, what I'm expecting to see, if it's negative for the toxin, is nothing. I don't even have anything to fight, right? It's going to be like this negative control. If it does have the toxin, it's going to give me this outward burst, this V type of shape, this appearance, because the antitoxin is seeping into this test strain and causing killing, let's say, of those bacteria, and it's causing killing of this toxin. So it's going to give me this type of configuration, which I can see on the positive strain as well. Is that clear?
Yes.
Yes. Okay, perfect. The person that asked first, is that clear? The dots. Yes, okay, perfect. All right. Now going back to the toxin itself, how does it actually exercise its effect and how does it cause disease? What it does is that it inactivates something called an elongation factor 2. Okay, it enters the cell and then it goes to this elongation factor 2 and just inactivates it. From the name, elongation factor, what is it supposed to do? It's involved in protein synthesis. It's like the elongase that just makes the protein longer so that it can actually be used and so that we can produce more and more proteins. If I no longer have this elongation factor, I will no longer be able to produce any proteins. Thus, I'm effectively killing the cell, right? So the diphtheria toxin enters the cell, it inactivates elongation factor 2, which prevents protein synthesis by the ribosome and eventually kills the cell. Is that clear? Yes. Okay, great. All right. This is our last slide before we move on to questions, which is the major concern with gram-positives and why they are such a big deal. Firstly, resistance. We have methicillin-resistant staphylococcus, which is very, very prevalent and unfortunately on the rise. I told you guys that this is also extending to include vancomycin resistance. So at that point, as your colleague mentioned, we would move on to linezolid, which is very sacred, and pharmacologists are trying their best not to use it, and they're like keeping it safe. We also have penicillin resistance, which at this point we consider super, super common. But penicillin used to be effective against almost every strain of bacteria. So penicillin resistance is on the rise in Saudi Arabia. Not only is resistance on the rise, but so is the spread of infection. So we used to only really be able to see those gram positives in healthcare associated infections or nosocomial infections. But now they're spread all over in the community. How many people have experienced strep throat? I know I have 1,000 times, right? Last but not least is vaccination. So although we do have the pneumococcal polysaccharide conjugate vaccine, it can only include a few of the multiple serotypes that are constantly evolving. So we're always going to have a lag there, and we're always going to lag slightly behind because we cannot possibly manipulate vaccinations as fast as bacteria manipulate their DNA. That brings me to our final checkpoint before we move on to practice questions. So if you guys have any questions for me at all, please feel free to ask them now. I would be more than happy to answer them. If you want me to repeat anything, then I will do so. Before we move on to practice questions, I tried my best to make them comprehensive and ask at least like one question about everything. They are only 10 questions though, so don't worry.
No questions on my part.
All right, great. Okay, I think we can go forward then. Okay, our first question. I'll give you guys time to read. Please type it in the chat whenever you're done. Take your time. I know it can get confusing. Okay, C. Yes, absolutely. You are both correct, mashallah. The answer is C because it's staph epidermidis, which is gram-positive cocci in clusters, right? It's catalase positive and coagulase negative. So staph, catalase positive. It's not aureus, so coagulase negative. Absolutely. All right, next question. Okay, that was very quick. Yes, absolutely. You are correct. Good job. Yes. So Staph aureus is involved in the pathogenesis of which of the following diseases? All of them. But it is asking about the super antigen specifically, which is going to be with toxic shock syndrome. Yes, absolutely. You guys are correct. Okay. B. Okay. B. Yes. Okay. I got both answers. Great job. It is Staph saprophyticus. You don't need to know the Enterococcus faecalis. It does not matter in this case, right? We do not care. What matters is sexually active, young female, dysuria, other symptoms of urinary tract infection. Immediately think Staph saprophyticus. Great job. Next. Okay, B. B. Yes, absolutely. The person that just joined, would you also like to answer the question? Or should we move on to the next one? I'll give you some time. I'll wait for just a few seconds. Okay. So it is B. Yes, you guys are absolutely correct. Because gram-positive cocci in chains, blood agar revealed beta hemolytic colonies, presence of bacitracin and hydrolyzed hippurate. And the whole point is, in this case, even if I don't really focus on all of those details, 2-week-old infant stopped feeding and became irritable, neonatal meningitis. Irritability, as soon as you see that, think meningitis. So what agent did I tell you guys? Ah, okay, I see. What agent did I tell you guys is the causative one for neonatal meningitis? Strep agalactiae. You guys are absolutely correct. Okay, halfway through the questions. This is somewhat tricky because you don't know all the information here, but there's just one keyword and you'll be able to get it. That's why I included this question. Okay, I have a C. Any other answers? Okay, it is C. Good job. So gram-positive rod is a gram-positive bacilli. So I'm starting to think, okay, this could be either uh, a Clostridium or it can be Corynebacterium, right? And then I'm starting to think, okay, it causes necrosis of tissue. Where did I see that? Gas gangrenes. That is tissue necrosis, okay? So this is Clostridium perfringens. Even though it was did not directly tell you that, this is Clostridium perfringens. This is gas gangrenes. So good job. All right. Next question. B. Okay. B. Yes. You don't even need to read the whole scenario, right? You see this bubbling type of appearance, which I told you guys is the H2O2 being broken down into H2O and O2. This is the catalase test. If catalase is positive, then it is E. Exactly. It's just like a fancy way of saying it. It's just to, because people are going to start looking for the S. So read the whole thing through. Yes, it is Staph aureus or MRSA, methicillin resistant Staph aureus in this case. Great. All right. Okay. MashaAllah, very quick answers. Yes, absolutely. This is A. So anaerobes, they lack sufficient catalase and superoxide dismutase. Yes. Okay. Okay. The first time that you guys give me different answers. No? Okay. Which one is it? Accidentally. Okay. Yes, it is C. So bacteriophage is going to be C. Conjugation is the process of the F positive sex pilus, right? And the conversion between a horizontal transfer, right? And last question. Okay, D. D. Okay. You both chose D. Unfortunately, this is your first mistake, and it's the last question. It's B. actually. They are short sequences of DNA. Transposomes are DNA. Remember how I told you guys that the plasmid DNA can either get reintegrated back into the chromosome or a portion of the chromosomal DNA can become a plasmid? It's all DNA. I'm not involving RNA at any point. Okay? Does that make sense? Okay. But the rest of the option D is correct. Yes, yes, they are. If it was DNA instead of RNA, yes, that would be correct. Good job. And with that, we come to the end of the session. It's been an absolute pleasure having you guys, and I sincerely hope that this session was beneficial. I was very, very glad to host it. Here's to many more, inshallah. If you have any questions or concerns, please feel free to reach out to me at any point. I'm always available to help, and I'm always more than happy to help. There are no such things as stupid questions or you bothering me. It's my job. Okay? Of course, I'm not sure who you are, but you're welcome.
Thank you, Farah.
Thank you
Genuinely enjoyed
guys.
hosting your session.
Thank you so much. Is this